Hello, you're listening to BioViews, perspectives on the life sciences across time and place. Welcome to all of our listeners tuning in today to the very first double episode of our podcast. I'm Laura Ruggles, and today we're going to explore the world of ancient DNA research. My own first encounter with this field, probably like many of you, was at the movies as a child when Jurassic Park first hit the screens, full of reckless scientists bringing dinosaurs back to life using DNA from ancient mosquitoes trapped in amber. Fast forward several decades, and while we don't yet have dinosaurs roaming the streets, the field of ancient DNA research is growing rapidly. Today, to guide us through how scientists study ancient DNA, we'll first hear from Dr. Elizabeth Jones. Liz is a historian of biology at the North Carolina Museum of Natural Sciences, and she's going to tell us a bit about how the field of ancient DNA research first emerged. Later in the show, we'll be joined by Dr. Laura Wyrick. Laura is a paleomicrobiologist at ACAD, the Australian Centre for Ancient DNA at the University of Adelaide. She's going to share a little about her own work and give us a sense of what ancient DNA research looks like in practice today. Recently, Liz published a paper entitled Ancient DNA, A History of the Science Before Jurassic Park. She Skyped in to talk with me about what ancient DNA is and what the field looked like in the early days of ancient DNA research.
Sure. So ancient DNA research broadly is the practice of extracting, sequencing, and analyzing degraded and damaged DNA that comes from ancient and extinct material that's anywhere from tens of hundreds to thousands of years old. And ancient DNA has been extracted from different types of organisms, from plants and animals to humans to even bacteria. And it can be found in substances like tissue and skin and even bone. And then fossil material as well, so long as the fossil is not completely mineralized yet. So how did the field get started? Well, ancient DNA is a relatively young practice considering the history of science overall. You could probably put its start date around the 1980s when scientists started thinking about the question, okay, can we get DNA from old material? So at that point, they knew they could get DNA from modern material, so living plants and animals and humans, but they hadn't really started thinking about how far back in time can DNA survive. So that really started, I'd say, the late 1970s, early 1980s, when you had a variety of scientists starting to think about these questions and actually start testing them out. So seeing if they could get DNA from really old material. And they started with some really old specimens like multi-million-year-old insects in amber. They also started with frozen mammoth bones. And a lot of this research, in the lab at least, was conducted at the University of California, Berkeley, with a lead researcher named Alan Wilson. But I think it's important to note when we're talking about the history that these ideas of extracting DNA from ancient and extinct organisms actually arose elsewhere. So there were other characters like a man named Charles Pellegrino, who was a paleontologist, who started thinking about the idea of, can we extract DNA from insects in amber? The same idea was kind of thought of by a different individual named John Taklash. He's a paleontologist enthusiast, but really a dermatologist by profession. And then there's also George Poinar, who has a lot of connections with Michael Crichton in the Jurassic Park book. So I guess the point is that these ideas about can we get DNA from old things, from dead things, had its origins in around the 1980s, but it didn't start in really one place at one time with one person. These ideas were popping up in different areas.
As well as all this interplay between independent people coming from very different perspectives that all began to converge on this one idea that we could extract DNA from ancient organisms, Liz really emphasised the practical and public dimensions of the field's emergence too.
One of the fascinating things about ancient DNA research and studying it as a science, and also the history of it as a science, is that it was always a public affair. Science in general doesn't happen privately and in the lab. Yes, there are components that are certainly private, but for ancient DNA research in particular, it was always of interest to the media and then to the public. And these ideas were inspired outside of the lab, infiltrated into the lab, and there was this really neat interchange between the professional and the popular. And so that's what the history of ancient DNA is. So that's what that paper really shows is from the beginning, you have the seeds that are planted. The ancient DNA is very much of interest to the media and to the public. And there's a lot of speculation and there's a lot of spectacle. And those features were really fundamental in helping get ancient DNA, which was not at that point really an established practice, on its way to becoming something that people knew about, something that researchers could go and do and contribute to. And I think understanding that from the very beginning helps us understand why ancient DNA is so popular today and why movies like Jurassic Park still capture the public fascination, but also still more importantly, influence the science and propel it forward as well.
A lot of the roles of public and media engagement and speculation at the time that she talked about sounded very positive, raising interest, propelling the field of ancient DNA research forward. I wanted to know whether there are any negative impacts or unreasonable expectations caused by this sort of publicity too.
Yeah, that's a great question. And it's one of my favorite, actually. My position is to be hesitant to say that something has had a clearly negative or positive impact. My job as the historian, I want to say, also to be a bit diplomatic, is to say that it's had an impact, and its positive or negative impact is how the scientist or how someone else in the story that I'm telling perceives it. So yeah, 100% ancient DNA's really close connection with the media and with the public and with Jurassic Park has had, according to scientists' perspective, both positive and negative influences. So, its positive influences, there are clear links to increased funding, there are clear links to increased publicity, which leads to student recruitment, researcher recruitment, and publication timing, and other things like that. So you see that there's this really neat interplay between science and the media and the process of science. So what gets done and how it gets done. But then there are also scientists that think there are negative influences. And a lot of that has to do, again, with the connection between Jurassic Park and the role of expectations. One great example in the history of ancient DNA research happened in the 90s, and it's one of the number one questions that ancient DNA researchers get, that I get. Well, can we get dinosaur DNA? Or how far back in time can we really go to get DNA? The answer's been there for about 20 or so years, but it's still one of the most popular questions because of movies like Jurassic Park. And in the 1990s, you have a series of research teams who actually tried to get DNA from either dinosaur bone or DNA from insects in ancient amber. You have about 10 years of back and forth research saying that yes, we can get this DNA that's multi-million year old DNA. And then you also have research questioning it as contaminations. It's environmental contamination, it's modern contamination, and in one case, the DNA that was extracted from dinosaur bone was actually shown to be the DNA from the human researchers in the lab at the time. So for a lot of the ancient DNA community, that was a huge, they would think, embarrassment because they had to backtrack on something that they had pushed out to the public. The issue there is that a lot of scientists thought that this was really bad for the field. And yes, in a way, they had to do a lot of damage control. But at the same time, I think it's a really great example of the process of science, doing the research to figure out what's possible and also what's not possible. The interesting thing with ancient DNA is that all of this happened on a public playing field. So it wasn't a private affair. The dinosaur DNA was broadcasted across the media. It was published in one of the top journals, Science. So damage control happened on a private and a public front. And that's where a big mismatch of expectations became really problematic for scientists, but also it was very much a story for the public about we tried something and it didn't work and here's why. It's a double-edged sword. It helps them speak to the public, but they also have to be really careful about the nuances, where the differences lie between what the movie says they can do and what they actually do on a day-to-day basis.
I reflected on how these sorts of missteps are something that we often actually miss in the stories that get told about science, and are a bias in scientific publishing generally too. We hear the results of the success stories, and not often the long, messy process by which they were arrived at, or the many dead ends that were important to figuring out what is not the case, what we can't do, what methods don't work. These are parts of the scientific process that is just as crucial as those wow moments when we get some important positive finding.
Yeah, exactly right. And you the public would see a different history of the science. Were that the case, like you said, if those negative results were published. And in the history of ancient DNA, it's kind of nice because a lot of those, what are now considered negative results, were published. So we have an interesting track record of that, which in other sciences, sometimes you don't have. So, yeah, that's a great point that you bring up about a different kind of story or side of the story.
I asked Liz if she could tell us a little bit more about some of the technologies and methods that were both driving and limiting ancient DNA research during the early days.
Yeah, so technology plays a huge role in the history of ancient DNA research and how researchers look at the technology and try to figure it out to adapt it to their needs when it comes to something like trying to get DNA from fossil material is really important. So when we're talking back in the 1980s, there was a technology called the polymerase chain reaction, which is abbreviated as PCR. At the time, it was primarily used for the extraction of DNA from modern material. And that's when scientists thought, well, maybe we can apply it to fossil material. The thing is that the amount of DNA in fossil material is so small and it's often so degraded and damaged that it's really hard to use that technology to capture it, to pull it out, to amplify it, to make it in a way that we can actually study it and have enough genetic information to make sense of it. So that way it's actually useful for the kind of evolutionary questions that these scientists are asking. So a lot of what researchers were having to do was kind of test what substances, so what kind of fossil material better preserve DNA, higher quantities and also higher qualities of DNA. This technology was really, it was kind of like trial and error throughout the 1980s. What worked, what didn't work. And the question they were asking here sometimes wasn't so much of an evolutionary question or a biological question. It was very much theoretical. Like, what can we do with this technology? How much information can we get? And under what conditions does DNA better preserve? And then as the 30 years go on from the 1980s to today, the technology progressively gets better. Doesn't mean that doesn't have problems. So with strides, you also have some setbacks, and you're still trying to understand kind of artifacts and the role of contamination.
I found it really interesting how now, even though the techniques and technology are becoming more sophisticated, a lot of these same sorts of questions are still really pertinent for ancient DNA research today. Liz spoke about so many strands coming together to drive the development of this field: the public, the individuals, movies and media, technologies, and methods. I wondered whether this history of ancient DNA research could tell us more generally about some of the factors involved in the development of new fields and other research programs, or whether ancient DNA was special in some way.
Yeah, that's a really great question. I feel like there does need to be some more research done about how the press and the public play a role in the development of science more generally, not just in the case of ancient DNA. My opinion is that ancient DNA is unique in some ways, but that when we look at sciences, particularly when we're looking at the development of much more recent sciences, we should always be looking at the role of the press and the public as being a crucial component in that, because I think that what we're seeing today historically is that modern media and other developments like celebrity culture and the science communication movement are playing a real role in the process and the practice of science. And that doesn't just go for ancient DNA, it goes for all sciences. Funding is a very real issue, and science communication is a very real issue. And a lot of scientists today are expected to communicate to the media and the public in order to keep their funding, to get the grants, to get the research. to keep their jobs. It's a part of the process. So science is not immune from the economy. The press and the public are a link from the lab to the funding sources. And what we're seeing in ancient DNA is that ancient DNA researchers are really savvy at capitalizing on celebrity and using it to their advantage. And I think that this is really, and again, I don't want to say it's a positive or a negative aspect of the science. I want to say it's a very real one. And for me, it seems like it's very pragmatic. And I imagine that there are other scientists who are equally savvy or are becoming more aware that it's important to be savvy with the press and the public for these kind of purposes. I think the message more broadly for other historians of science and sociologists of science and scientists themselves actually is to look at the bigger picture and see what kind of other movements in modern media and celebrity culture are going to be useful for them as far as getting done the work that they want to get done, but also how these movements present new challenges and how they infiltrate back into the process of science. So it's not just that the science of ancient DNA inspired a great movie like Jurassic Park, but the point is to also look at how a great movie like Jurassic Park, a worldwide blockbuster phenomenon, has actually impacted the science of ancient DNA itself. And that's where that really neat interplay comes in. And I think there are other sciences, perhaps like astronomy and AI, artificial intelligence, those kind of other areas of research probably exhibit a similar interplay between science and the media.
That was Dr. Elizabeth Jones talking about the history of early ancient DNA research. Joining us now for the second half of the show is paleomicrobiologist Dr. Laura Wyrick, who's going to talk to us more about what ancient DNA research looks like in practice today. Having read Liz's paper on the history of early ancient DNA research, I asked Laura whether she had any initial thoughts on the article or on ways that understanding the history of their fields might be useful for scientists.
The article was incredibly interesting, and I haven't worked in the ancient DNA field for all that long, just since 2012, but the paper really taught me a lot about what I thought I already knew. So certainly we have to realize that these fields can be much longer than just the time that we're actually working within the field. And I think there's a very common sort of approach in recent era to really look at references that are recent and not really go back into time and use this history of science to look at how this field was established and what the questions were surrounding the basics of the field that you work within. Certainly, it was very refreshing to go back and read where these researchers started from and to look at all the sort of hard times and all the criticisms that they had to face to even get this field off the ground. It was also fascinating to read about how similar the field still is. We're still worried about contamination, and we still don't have a dinosaur sequenced, and we probably never will, but that's another story. So I think, in reading this paper, it really caused me to think about the basics of what we're doing and how we've gotten to where we are 30 or 40 years later. I still think we have a long way to go, and I'm glad to see that some of the early frustrations in the field are still things that I'm frustrated about today. I don't feel so alone. But it's absolutely sort of underpins the basics of everything we know and really causes you to go back and think about your own research and refocus it in a unique way.
And clearly, we don't have dinosaurs or anything roaming the earth again, but in the public imagination, resurrecting extinct animals is still often what people think of when they think ancient DNA. I wanted to know what kinds of things ancient DNA researchers are actually looking at today.
Absolutely. So, Jurassic Park was one of these fantastic, super cool books that came out, and everybody thought, wow, we can recreate dinosaurs. There's been a lot of research that's happened since then that suggests the theoretical survival and half-life of DNA is only about 1 million years. So the oldest ancient genome to date is 680,000 years, around in there, maybe 700,000 years old. There's a little bit of error with the dating, but that's really a far cry from, 30 or 300 million years or 500 million years, some of these places where we'd expect to see dinosaurs more prominently shown. And so we really don't think that we could probably get DNA out of some of these specimens because we think the DNA will have literally broken in half so many times that we physically can't pick it up anymore. The story of different cellular molecules and things like that being preserved in amber is an amazing one. And there's been a lot of papers out in the field that have tried to get DNA from out of amber, and it's just failed time and time and time again. So even though that idea of getting DNA from inside a mosquito that bit a dinosaur sounds like super cool, science, it really hasn't proven to be true in the field. And so the last, especially 20 years and even 10 years of research have really shown that it's really not true. Most of the DNA we get out of amber these days is bacterial from contamination from the people who have been working on the samples. And so even if they were trying to do it in the 80s, they probably contaminated some of the samples so much so that we wouldn't be able to have a proper look at them today. Ancient DNA is super broad, so you can look at anything that's biological. So we can look at humans, animals, microorganisms, viruses, fungi, you name it, we can look at it as long as it was living, and living relatively recently compared to dinosaurs. So ancient DNA just refers to DNA that's preserved within biological specimens that weren't intended to be preserved. So this would be things that had died naturally in the environment, things that weren't sort of stored in formaldehyde for a medical purpose later in life. But it's really, really broad ranging.
I asked Laura to tell us a little bit about the kind of research that she does.
So I'm a paleomicrobiologist, and I work on looking at ancient microorganisms, whether or not that's bacteria, archaea, viruses, or even fungi. So we're really interested in how these microorganisms exist as a community and how they change through space and time. Whether or not that's an adaptation to disease or to diet or to changes in the environment, we're interested in it all. And we're also interested in comparing those microorganisms to other information that we might see. For example, the human genomes from where those microorganisms may have been obtained. So we can really sort of start to look at how a human genome might sort of influence the microorganisms that person's body would also select for. So the bread and butter of our research is done looking at calcified dental plaque or dental calculus. It's that sugary slime that you feel on your teeth after you drink a bottle of Coca-Cola or something. And it's really a bacterial biofilm that's growing and multiplying in real time. And if you don't brush your teeth or you don't go to the dentist, it calcifies into a rock-hard matrix. And that preserves extremely well along with the fossil record. So it really provides a way for us to go back into time and to look at sort of what microorganisms people had and what they were carrying with them. We can also do this in animals as well. So it's a similar process, even though I wouldn't necessarily call it dental calculus. But it really provides a window into the past about what sort of bacteria were in different human populations and what was there.
Liz talked a bit about the sorts of things that were driving the early ancient DNA research program. I asked Laura what kinds of factors or technologies are driving the leading edge of research now.
That's a fantastic question. So a lot of our research is driven by where we can get funding. That's, flat out one of the games of being a scientist in the modern era. But a lot of from the technological side, a lot of our science has been driven by the advent of next generation sequencing. Instead of looking at one DNA molecule, now we can look at millions and gosh, even sometimes billions of DNA molecules that are present across different samples and different individuals. And so that really changed the game in giving us better resolution and better sort of clarity into what we were looking at. She talks a lot about contamination within this paper and how some of the early people really did a great job of looking at that, and others didn't, which she actually doesn't talk about in the paper, which is interesting. Svante Paavo very famously, supposedly sequenced his own DNA in that early mummy paper instead of the mummy itself, but that's not mentioned in the paper, which is interesting. Anyway, it's one of those things where technology now allows us to tell the difference between contamination and what's potentially real. We get enough DNA sequences and we can actually look for patterns of damage and patterns of breakage that would suggest it's ancient in nature. So that's really fundamentally changed the field, this ability to sequence millions and millions of DNA molecules. But that said, I really think that social structure and anthropology have really shaped our field, especially mine and where it's headed. I'm also a microbiologist by training, and so microbiology is shaping how we sort of view ancient DNA and the information that we can get out of it. These social sciences are really sort of allowing us to understand what these changes mean in a better context. Microorganisms, for example, this sounds bizarre, but are extremely adaptable to changes in environment or diet, but those are really cultural changes when we're talking about ancient humans. Those are really changes in lifestyle, changes in religion, changes in practices that can be related to much larger social constructs that are not necessarily just environmentally related. So it's a really interdisciplinary field right now. You have to think about, techniques and hardcore chemistry all the way through to sort of anthropology and religion in some cases. And so it's a very, very diverse field and requires scientists to be on their toes all the time.
Laura mentioned funding as an issue. Given the expense, differences in local and national economic situations, and funding sources, I asked her whether this meant that in practice the distribution of where this kind of research even could be done would be quite patchy.
Yeah, ancient DNA work is incredibly expensive. So it's on par with, say, cancer biology. I mean, we really still have to expend, incredible amounts of money to get DNA from ancient samples. And so, as an anthropologist, you're not going to get the same level of funding as you would if you were a cancer biologist, for example, to do the same sort of research. And a lot of that has to do with societal outcomes and what's weighted as being more important. So certainly there are samples that we'll try to go after within the field that only contain even 1 to 5% of true or endogenous real ancient DNA, and the rest of it can be contaminants. And so that means we have to sequence that sample that much more to get what would be just coming out of one simple modern sample. So for us, it's incredibly expensive to do the DNA sequencing. You're talking thousands of dollars per sample. And so that really sort of starts to stratify research based on countries that are willing to fund large amounts of money for anthropological or archaeological questions. And so I don't know if it's woven within human fabric or human knowledge, but the countries that care much more about their history and where they've come from and appreciate those sort of things have in history tended to fund ancient DNA research much more aggressively than some countries. Certainly there's been quite a few write-ups in the United States kind of supporting American scientists that have really been struggling to get funding for ancient DNA research. And that's often because much of the archaeological research is funded at a max of $300,000, $400,000 per grant, whereas medical grants will be $1 million or 1.5 million or something along those lines. And so considering your research costs the same amount, but you don't have the same amount of money, means that you're very stunted in what you're able to do. So certainly there's a social and an economic factor that play into our ability to do ancient DNA research and do it well.
I wondered whether this research of an archaeologically and anthropologically integrated sort goes on in Australia, working with Aboriginal communities here in ways that could help them reconstruct their histories.
Absolutely. So at the Australian Centre for Ancient DNA here at the University of Adelaide, Ray Tobler is leading a really big effort to reconstruct ancient Aboriginal Australian and Torres Strait Islander genomes, as well as mitochondrial genomes, with the idea of tracing back their evolutionary history and understanding where people were and for how long in different parts of the country. And they work directly with the communities to do that. So they go on country, they have discussions with the communities about the research, and they actually get sign-offs from the ancestors of those individuals, of the samples that they're going to assess. They go then and do the research, and then they go back to the communities and they report that information, they discuss it with them, and they're really trying to incorporate songlines and storylines and history into the sort of ancient DNA analysis that they're doing. So absolutely, that's happening here in Australia. For my team specifically, we're also trying to look at the microorganisms that lived in ancient Aboriginal Australians and Torres Strait Islanders as well, to really understand what the health was like before European arrival and how we might sort of reconstruct some of that health to benefit Aboriginal Australians. But really sort of, that's starting from the ground up as well. And because we want to do these projects in conjunction with the communities, it does take a long time to get them going, but we wouldn't have it any other way.
Laura talked a little about how technology helps her team to figure out what part of what they've sequenced is the ancient animal or human DNA that they're trying to get at, and what parts are contaminants. I asked if she could tell us a little about how modern researchers try to minimize contamination of the samples in the lab as they're working.
Yeah, I think the best analogy is to think about those outbreak movies where you've got people in complete hazmat suits, where they only have, a little bit of their eyes that are observable and everything else is in a bright blue or bright orange suit. That's basically how we work on a daily basis. So we have a very specialized ancient DNA facility here at the University of Adelaide, and everybody that goes into it has to be highly trained, so about 6 months of training before you can go in and work by yourself, and you work all day in one of those suits. And so the idea behind that is that you are the biggest source of contamination that you could possibly ever introduce into your samples. So you basically exclude anything from your body going into the lab. So you basically just wrap yourself in this suit, and that prevents at least most of the DNA from you going onto your samples. But within this laboratory facility, we also have things like UV irradiation, HEPA air filtering, and regular cleaning with bleach to break down or limit any sort of DNA from coming into the laboratory. So the idea there is to really just limit any of the sort of exposure to modern DNA that sample could have. So we also, despite that, have bacteria and microorganisms that like living in those conditions. They like living with bleach and ultraviolet radiation. And so we've actually selected for a very unique set of microorganisms that live within the lab that like those conditions. So despite all of this, work, we still have to account for these weird microorganisms that also live in the lab when we look at our data. So we also include controls that are just, if we're doing a DNA extraction, for example, it's an empty tube that we just run through the exact same DNA extraction, which sounds a bit crazy and a bit, worthless, but it's actually picking up all the DNA that's just present in the lab. So it's picking up this microbial signature of bugs that just live in this weird lab environment. And so we can account for that in our analysis when we get to the end. But what that doesn't account for is everything that sample's been exposed to prior to the lab. So, for example, one of the things in the field to identify a rock from a bone is actually to lick it, because they taste very different. And that really allows you to say, oh, okay, this is a bone, not a rock. The problem with that is that directly deposits all of that archaeologist's oral bacteria onto the sample.
Yeah.
And so if they didn't realize what they were going after, if they didn't realize they were dealing with a human remain, they could have put their hands all over it, they could have sneezed on it, they could have eaten their lunch over top of it. And so there can be lots of modern DNA that's exposed, on the surface of these samples. So we also do a really nice job of decontaminating the samples and trying to remove any of that outside surface to try to remove any of that DNA that could have been introduced prior to the sample arriving to us. And last but not least, we just do a lot of comparative genomics. We compare our samples to the sort of profiles that we see in the environment or in other labs or from other humans to really try to understand where our signal is coming from and why.
I asked Laura for her thoughts on the future of paleomicrobiology and what kinds of developments are on the horizon.
Yeah, so far we've been able to reconstruct ancient microbial communities from Neanderthals, and the oldest community that we've constructed to date is 48,000 years old. So I think new technologies will allow us to go back even further into time. Looking at even smaller DNA fragments, being able to effectively identify them will happen. So we can maybe push up to 100,000 years or a couple 100,000 years of ancient oral bacterial species we can identify. The other thing that really may come into play here is starting to identify ancient viruses and things that are much more difficult to identify, even in modern space, to really look at those within the ancient record. But this all really comes down to hand-in-hand relationships across anthropology, archaeology, microbiology, and molecular biology to really sort of get these teams working together, because an increased collection of the information and what we call metadata, the sort of data around the individual you're studying, despite all the molecular biology we do, actually becomes very, very critical in us being able to effectively interpret the results that we see. So yeah, I think moving forward, we need to be more interdisciplinary, and we need to rely on increased, chemical and technological advances that will allow us to look at shorter and shorter DNA fragments.
Those large collaboration-enabling data and metadata sharing initiatives that Laura mentioned sounded really interesting. To finish up the show, I asked whether she could tell us a bit more about how they work and how data gets shared around the world.
That's a great question, and one that is constantly evolving within the field. It's really our practice and our mindset right now that what you would do is create an ancient DNA data set, and then you would not only provide that data to anybody who's interested, but you would also provide that metadata about those individuals. And on top of that, you would provide the analysis pipeline, the exact bioinformatics scripting and code that you use to interpret that data, along with all of that, so that somebody can directly reproduce your results. And so there's a couple different ways that can be done. One of them is hosted here at the University of Adelaide. It's called OGRE, the Online Ancient Genome Repository. So you can go on there and you can look to see which genomes, ancient human genomes and ancient human microbiomes, these collections of microorganisms, have been deposited from all over the world. And so I think we're pushing up to 1,000 ancient individuals now at this stage, and it's really, really exciting because it's a place for people to share their data and share that analysis tool and share that metadata in a way that we can now do cross-cultural comparisons all over the world. But this is just one of many. I know there's a couple more coming out, so we'll have to keep our eyes peeled.
Thank you so much again, Dr. Liz Jones and Dr. Laura Wyrick, for joining us on BioViews today, and thank you all for listening in. Don't forget to tune in again next fortnight, where we'll be hearing all about consciousness and creatures from the deep. Subscribe to our blog here on this page or follow us on Twitter at BioViewsHPS. You can also access Liz's article, further reading on ancient DNA, and follow the research of today's guests by using the links on this page. That's all for now till our next episode. I'm your host, Laura Ruggles, and you've been listening to BioViews.